My name is Rich Phillips, in case it's been so long you forgot my introduction. Today, I'm going to tell you about some of the work that we've been doing in our lab, where we've been trying to understand how forests function and trying to think about how very fine-scale interactions happening at the level of the root interactions with their mycorrhizae and their microbial communities interact larger scale forest processes. This is a picture high atop our flux tower that we have in Morgan Monroe State Forest, where you can get a really nice view of sort of the forest. And this is what I think many of us think of. But what I'm going to hopefully convince you of today is a lot of the interactions that are happening below ground, things that we don't see, that are not necessarily part of this macro level view of the forest, are really important for things that we care about in terms of application and forest function. So I like to start by reminding you about why we care about forests and forest functioning, and a lot of it has to do with climate change. This is a plot of the amount of carbon dioxide emissions that we've been releasing to the atmosphere over the last 150 years or so. And you can see the emissions are dominated by fossil fuels and obviously to a lesser extent land use change. And if we think about where all that CO2 goes, you can actually do a mass balance at the global scale. Think about who is responsible for taking up and removing that CO2. So about a third of it goes into the oceans. About a quarter of it goes into our forests or to land, but mostly into forest. And then the residual, that little blue part that's in the middle there, this is working. This right here, this atmosphere part, this is the stuff is the residual, and that's what traps heat. So anything we can do to increase the amount of carbon uptake in the land or in the oceans means less that goes into this blue box, which can basically slow climate change. Now, the challenge is with the ocean, there's much more ocean than there is land, but you can see it's pretty constant amount of uptake by the oceans, and that's mainly because it's CO2 solubility as a process of physics. It's not necessarily a biological process that drives that. The land carbon sink is highly variable, and that's because it's very dynamic. All the different things that affect the amount, what I'll refer to as the land carbon sink, the amount of CO2 that's removed by land ecosystems. But that also presents a lot of opportunities and challenges for thinking about how we might increase the amount of carbon uptake by the land and slow the pace of climate change. So one of the challenges that we have to understanding how and why and under what conditions the land sink is operating is that a lot of it's controlled by the availability of nutrients. So one of the biggest uncertainties is what role nutrients play globally in the land carbon sink. So one of the efforts to unpack this was a big inter-model comparison. They're called CMIPs. This is where all the best modelers in the world sort of get together with their models. They run their models with the same climate forcing data, and they try to make predictions of whether the land will continue to be a carbon sink in the future. And as you can see, everything below this sort of zero line is the land acting as a sink. But as you get into the year, past the year 2024 or so, you start, or 2023 in this case, you start to see wide model divergences in terms of whether we're going to continue to have a carbon sink at all. Maybe we'll have carbon sources from our land. Now, these early models had no nutrient limitation. There was no way in the model to actually represent nutrient constraints to productivity. They're largely driven by they're photosynthetically driven models, or mostly driven by water and light availability. And recently, in CMIP5, a group of these models started to put nutrient limitation into the models with the idea that maybe this will help us and sort of narrow the scope of these different model projections. Again, each of these different colors is a different model looking into the future, trying to model the land carbon sink, and it didn't really help. And that's because it's very difficult and challenging to model nutrients. And I would say the key challenge with modeling nutrient availability is that what controls nutrient availability are the interactions between roots, microorganisms in the soil, and their interactions with minerals that basically are the key drivers of what determines this nutrient availability. And we don't have good representations or good ways of modeling this right now. So what do we do about that? Well, one way, maybe this is a way that's familiar to a lot of you, is we think about trying to model or conceptualize the heterogeneity of plant-soil interactions. There's hundreds of thousands of tree taxa in the world. There's limitless number, millions or more of microbial taxa, OTUs, ASVs, however you want to define it. And so sort of figuring all that out is very difficult. And so what we often use is just simplifying frameworks where we look at traits. So for example, we might look at how leaf traits and wood traits interact with root traits and how these lead to different gradients. And you have a conservation gradient or a collaboration gradient. And these sort of conceptualizations are a first pass at really trying to understand the diversity of form and function of plants and how we can start to sort of think of them in concrete ways to make some predictions about, the land carbon sink. But really the holy grail to all of this, I would argue, is not just understanding traits and looking at the plant traits, but it's looking at the interactions between plant traits, the characteristics of soils and the soil microorganisms that live therein, and the microbes that live within the soil. And so that sort of intersection really is primarily what we're interested to understand these biogeochemical syndromes, or what I often call an integrated ecosystem phenotype. What's something that we can think about that maybe integrates across all this heterogeneity and gives us something, a unit of measure or of granularity that is useful for both making predictions and for understanding process. And so with that, we hope to do that to understand ecosystem functions, services, and resiliences to environmental change. So that's the backdrop of what motivates me. So how do we go about doing that in my lab? My lab, we go about that by looking at the two dominant forms of mycorrhizal fungi that associate with trees. So nearly all trees on Earth associate with mycorrhizal fungi, and most of them will associate with principally with only one or the other type of mycorrhizal fungi. So maybe these are familiar to you. If you've never been to a mycorrhizal talk before, hopefully this is one take-home message you'll get from this, which is that there are great differences in the arbuscular mycorrhizal trees that associate with arbuscular mycorrhizal fungi versus trees that associate with ectomycorrhizal fungi. Arbuscular mycorrhizal fungi are more ancestral. The ectomycorrhizal are derived from saprotrophs. They've evolved independently something like 80 different times. They have much more large and striking differences in both morphology and function. But for the purposes of this talk, I'm just going to tell you about two things that are different, which is that for the most part, the AM fungi do not have the ectomy to make extracellular enzymes, which can break down organic matter and liberate nutrients for their symbionts, whereas our ectomycorrhizal are more, and we consider them more scavengers. The ectomycorrhizal are more miners. They have the ability to make these enzymes to make oxalic acid and other things that can actually remove nutrients from organic matter, and in doing so, provide those nutrients back to the host plants. Now, throughout the talk, I'm going to use these symbols. They happen to be red and blue. I realize this is a swing state and an election, this close to election. Please don't read anything into, there's going to be a lot of maps of red versus blue. Just, it's totally coincidental. Now, here's a map of red and blue and some other colors. This was a recent idea of, if we want to understand how AM and ECM fungi may be influencing different processes, we think about where they exist on the landscape. So, for example, in this map right here, the red is the areas where the particular type of mycorrhizal trees are dominant. So on the left side here, you can see that AM fungi, or trees that associate with AM fungi, are dominant in low latitude ecosystems, mostly tropical forests, whereas ectomycorrhizal associating tree species are mostly dominated in high latitude ecosystems. And this makes a lot of sense given what I told you about their enzymatic capabilities. If you're to live in a cold and wet or cold and dry environment in a high latitude region, having the ability to mine nutrients out of organic matter is obviously a very successful adaptation that you might have. Now, in the AM, being able to scavenge where things are warm and wet and things are decaying really fast, that's probably an apt adaptation for those environments. But what I'm going to talk about today is what I think is a more interesting question, is how do they interact in the temperate forest? These temperate areas where both of them co-occur. So the green-yellow just means they're about near even abundances in temperate forests. And there are obviously hotspots across the landscape, but it's a really good place to ask this question about mycorrhizal function, and that's what I'm going to focus on today. And so the question that I sort of asked throughout this talk is, can mycorrhizal types, the trees associated with AM versus ECM fungi, be integrators for a suite of other characteristics that can help us understand forest functioning? So just a quick roadmap for the talk. I asked this question about plant-microbe-soil interactions. Do they differ between AM and ECM trees in temperate forests, and what are some of the consequences for ecosystem dynamics? And then how can this framework actually help us understand responses and feedbacks to global change? And the way I'll do that is sort of toggling back and forth. This is the way we do things in our laboratory. We do some observational work, we run experiments, we do mathematical modeling and synthesis and meta-analysis, sort of kind of constantly toggling back and forth between all of those, developing inferences, but also new questions and hypotheses that we can address with other modalities. Okay, so you're all up here in Michigan, so it's not, I don't have to tell you where Indiana is, but Indiana in many places is the perfect place to ask these questions that I'm about to talk about today. Why is it perfect? Well, we have amazing parks. You probably know about some of our parks that we have in Indiana. We have really strange things that occur in our state. And we actually have, what makes it unique is, in more seriousness, is that we have a lot of secondary forests, probably similar to Michigan. This is actually our state seal of a guy cutting down the forest here as the bison roam. But we have a lot of secondary growth forests, about 85 to 125-year-old forests that are sort of blanketing, especially in the southern part of the state when we're talking about. And I don't know how this slide got in here, but I guess we're known for our football team. That's what some people tell me. So the real reason that we study Indiana forests and why it's such a good model system for us is we have near even abundances of AM and ECM trees. So we have dominant tree species like sugar maple, tulip poplar, sassafras, cherry. We have some ash left, not many, elm and black walnut. So those maybe are familiar names to you. Those all associated with AM fungi. And then we have red oak, pignut hickory, white oak, American beech, many other oaks as well, shagbark hickory. We also have some gymnosperms. Those are all in the Mycorrhizal-Associated tree species. And because we have them co-occurring as canopy dominants, it's a good place to ask some of these questions about AM and ECM trees and their effects on the landscape. So our hypothesis going in here is that given these sort of differences in enzymatic activities, that these AM-associated trees will have different nutrient use traits, other characteristics of the plants that sort of map onto these different fungal strategies. And that might lead to, because they have different fungi, they have different traits, potentially different nutrient cycling syndromes where AM and ECM trees co-occur or are dominant. So the first pass that we want to take is we want to look at, let's take a bunch of trees associated with AMF, a bunch of trees associated with ectomycorrhizal fungi, and just look at the differences in characteristics of the trees. So Megan Midgley was my first PhD student, and she decayed AM or ECM leaves in these litter bags, sort of a classic way that we look at the decay rates of leaves, put out these bags in the soil, we bury them and measure them, the amount of mass loss after a period of time. She had 6 species, and Megan found this pretty striking pattern where on average, the decay rates of the AM leaves are about twice as fast as the ECM leaves. That was only 6 species. It was a field experiment. She did a lot of cool reciprocal transplants, but it was just getting us started and thinking about this. What other things might be different? So Matt Craig was a grad student, wanted to pick up on this. He decayed leaf litter. He did this in a jar because he wanted to use many more species. And what Matt found was like the most striking figure that I've ever seen with respect to looking at AM and ECM differences. These are the species down on the x-axis here, just ordered from left to right in terms of their decay rates. And as you can see, all of the species that decayed really fast were the AM tree species, and the ones that decayed slow were the ECM species. And so Matt did some chemistry and looked at why these leaves might be different. Turns out the AM leaves have more nitrogen and less lignin, which is not surprising given what we know about leaf litter decay rates. So that was cool. It tells us something, maybe we're on something. These are all the species here that collected from Lily Dickey Woods, which is one of our forests that's near campus. So these are all from trees on campus or near campus. Now, leaves aren't the only things, the only trait that sort of might affect the soil. So Katie Beidler decided she wanted to look at root decay. So she did a very similar experiment, ran it a little bit longer. And although her results were not exactly quite as remarkable as Matt's, that all the AM trees decayed differently than all the ECM trees in terms of root litter, she did find that on average, there was much faster decay of the root litters from the AM trees relative to the ECM trees. Again, also owing to the chemistry of these leaves being generally more nitrogen-rich and lignin-poor. Now, what hopefully I've established is that these plants that are associated with these fungi, at least in these Indiana forests, have these different litter qualities, which may have some kind of a feedback on the soils that develop. What does that mean? So we went to the soil and we started measuring different process rates. So this is a measure on the y-axis here of net nitrification, production of nitrate in soil. So organic matter gets mineralized, and then that ammonium basically gets oxidized to nitrate. It's the mobile form of nitrogen in the soil. And we found nitrification rates, this was across 30 different sites, were much greater in the AM-dominated forests. So these are plots where all the trees, or the majority, 80% or more of the basal area, are AM-dominated versus ectomycorrhizal-dominated. So big differences there in the nitrification rates. And then when you plot this on a sort of a continuous level response surface, so this is, the x-axis here is ECM dominance to AM dominance. So think of the plots over here. These are 100% dominated by ECM trees. These are 100% dominated by AM trees. So that's why they're down here is just, this is just percent AM dominant. So it's 0 AM means it's 100% ECM. As you run, if you measure plots all across this gradient, you see a relatively linear response, which is that as you increase the dominance of AM trees, you have more of this nitrification, which tells us there's something about these enriched litters that are dropping to the ground that are promoting this process of nitrification, this tree mycorrhizal gradient. So what's happening? Well, Ryan Mashinsky was a postdoc who was really curious about this. And in addition to the differences in the litter quality, Ryan said, are there differences in the microbes that may be processing the litter that are causing this sort of nitrogen cycling syndrome? So Ryan went out and he used shotgun metagenomics, and this is at one particular site, different than the previous sites. This is at Morris Creek. And what Ryan did is he looked at the estimated number of gene copies, and what he found is these are just the different genes that are encoding for the different pathways here, that in general, there are about five-fold more nitrogen cycling genes in AM-dominated stands than in ECM-dominated stands. Now, this was bacteria, so clearly there's a lot more dynamic nitrogen cycling. The bacteria are sort of have the capacity to process this nitrogen, and so these are just sort of the nitrogen cycling transformations that are occurring. Now, interestingly, the only color here that's not significantly different is the end fixation pathway, which is for, N2 right here, the bottom one. So there was not greater end fixation. So there's not that there's more nitrogen fixation. This is free living diazotrophic nitrogen fixers that are in the soil, but there's just definitely more processing of nitrogen that's happening in the soil, probably as a result of this more rapidly decomposing leaf litters. And then Ryan additionally went out and he wanted to look at the transcripts to see if the genes were then informing and inducing these the production of, this is ammonium oxidizing archaea and bacteria on the left and right. And what he found is there are more transcripts being produced in the AM dominated plots than the ECM dominated plots from the ammonium oxidizing archaea and the ammonium oxidizing bacteria. So these are the ones that are actually doing the ammonium oxidation, very different in these AM and ECM systems. Again, these are just for the bacteria. Now, you might recognize this guy. Andrew was very interested in looking at the fungal communities, and through a different set of plots that we set up in Indiana at 3 different forest sites, Andrew started looking at this question of the fungal communities and how we can start to make some predictions about what variables we need to actually predict and determine the structuring of the fungal communities. So this is a thing that I pulled from a collaboration I did with Andrew, and also obviously with Chris and others here. Initially what we wanted to do is say what percentage of variance, right? This is a variance partitioning tool here. Can we get from looking at, if we want to just predict or use soil properties, pH, seed-end ratio, a bunch of other metrics of soil, if you want to understand site, and not surprisingly, as you go from right to left, depending on what you're trying to sort of sort and classify at the phylum level, you can do pretty well with just knowledge of the soil properties. Site, you get a little bit more predictability, but again, it's, as you go into finer granularity, it becomes more difficult to do that. But when you add in knowledge from these different stands, these are AM and ECM and mixed dominated stands. I think there was 45 plots or so. When you add in the percentage of trees dominated by AM, and then you add in the interaction term between AM trees and the site, because this was done at 3 different forests, we have really strong predictability. And so at the level of grouping these fungi just by their functional guilds into pathogens, saprotrophs, so it's sort of a coarse resolution, but it's actually really good for modeling where we're not necessarily interested in every OTU. This works really well. And so it tells us that there's something about these AM interaction with AM and at different sites that is good for being able to understand and predict functional classifications of the fungi. Now, Andrew did some other work to build on this. So this is from a series of different plots. The color schemes just represent the 3 different forests. And as you can see, doing that same x-axis that I did before, as you increase the basal area of the AM trees on the x-axis from 0 to 100. So again, these are ecto-dominated, these are AM-dominated stands. You see this really nice linear relationship in the richness of the fungal communities. This is of the saprotrophs. This is of the abundance of the saprotrophs, the relative abundance. And then I won't present them here, but we saw almost identical patterns for pathogens, and then the opposite patterns, meaning a negative slope for ECM fungi, meaning that as you increase the abundance of AM trees, you have a decline in the abundance of ectomycorrhizal taxa that you pick up in your sequencing. So this is a fun-guild approach where we've been all the fungi, OTUs, basically classify them based on whether they're saprotrophs, pathogens, mycorrhizae, et cetera. So this is pretty powerful for a couple of reasons. One, it gives us good predictability, but two, there's some linearity that these mixed plots seem to have this sort of mixed phenotype where it's in between AM and ECM. Not exactly, but to some degree. All right, so I started with this question of, do we have these predictable sort of things that differ between AM and ECM dominated stands? And I would say, to support our hypothesis, yes, there do seem to be some differences that relate to litter quality, nitrogen cycling, bacterial and fungal communities that associate with different tree dominance. And that's a very useful thing for our predictions. So just to summarize that, our AM dominated stands have fast decaying litters that accelerates nitrogen cycling, and that selects for nitrifying microbes and a diverse and abundant group of fungal communities. Our ectomycorrhizal stands have slow decaying litters, selects for more conservative nitrogen cycling, which constrains microbial functions and fungal diversity and abundances. Now, I haven't really, and this is basically forms the central part of what we've been calling for the last 10 years, the Mycorrhizal-Associated Nutrient Economy, which is this idea that a bunch of things hang together. Think about this triad that I hear, that knowledge of the trees can tell us something about the soils and the microbes, and that basically they sort of hang together in some kind of an integrated phenotype that's useful for prediction. That's the concept of the main framework. But what I haven't really talked to you is too much about the roots and how the roots are sort of the primary agents, and that's what we started with, was trying to understand root allocation. So I want to briefly bring roots into this story. So Tanya Cheek was a postdoc in the lab, and she wanted to look at fungal production. So she did put out all these little sand pillows. These are hyphal ingrowth cores where we spiked it with a little bit of organic matter, and we wanted to look at the degree to which hyphae and mycelium would grow into the bags in AM and ECM stands. And Tanya saw something that was actually what we predicted initially, which was the ectomycorrhizal stand. Again, now this is a case where the blue is greater than the red. They produced more mycelium than in the AM plots. Well, that makes a little bit of sense if you think about it. In a nitrogen-rich environment, the investment in mycorrhizae and in foraging is probably going to be less in the AM stands. The ectos are in this more N-poor, slow decaying litter environment. They're going to need to invest more in their hyphae, and then the mixed plots have that mixed phenotype. And if you look at what explains that, it's really the amount of biomass, the percentage of biomass in the bags, the amount of mycelial biomass is proportional to the amount of nagase activity. So nagase is an enzyme that's involved in the hydrolysis or the breakdown of an amino sugar, which is a byproduct of chitin. So we used to think of this as a pretty slow cycling form of nitrogen. Now we think of it as actually potentially a very important and not so slow cycling form of organic nitrogen cycling. It's recycling. A lot of the chitin is likely to be fungal biomass. So it's recycling dead fungal biomass. And as you can see, the more mycelial growth in the bags also was tracked nicely the amount of this enzyme production, the more breakdown of chitin that we saw in these bags. Now, this was neat evidence, but it's largely correlative, right? We're looking at the bags, the enzymes and the hyphae in the same bag. So we wanted to do an experiment. So Eddie Brostek was a postdoc, and Eddie said, all right, well, how can we shut down carbon allocation below ground as a way of actually testing out that these syndromes might be different and driven by the roots? So this is a forestry department. You're probably familiar with the girdling. Girdling is a technique where you disrupt the movement, you disrupt the phloem, and you just basically are stopping the movement of carbohydrates from the leaves as being transported down to the root system. So we're effectively closing off this pathway here. And our hypothesis was if these carbon allocation pathways are important, by shutting off that pathway, we would induce a bunch of changes in nitrogen cycling activity. So let me walk you through this figure. It just takes a minute. So on the y-axis, this is the girdling effect. So the girdling effect means we're measuring that nagase enzyme activity, that organic nitrogen cycling proxy. We're looking at the girdle plots divided by the control plots. So anything that's above the 0 line means that the enzymes went up in the soil and the girdle plots relative to the control. Obviously, as you can see, all of the plots are below, meaning nagase activity went down. And so when we shut off the amount of carbon going below ground, we basically shut down the cycling of this organic N pathway, this nagase activity in soil. And it happened to a much greater degree in the ectomycorrhizal tree species than in the arbuscular mycorrhizal tree species, telling us again that by reducing this carbon allocation, that clearly these ectomycorrhizal plots are using this below ground pathway to accelerate nitrogen cycling. So whereas the AM plots are just dropping enriched leaf litter and there's a lot of nitrogen in circulation, the ectomycorrhizal plots, which have slow cycling litter, have to have this other way to mine nitrogen out of the organic matter and potentially out of the dead fungal biomass. And that was sort of nice proof of that with this study. So as I said, the greater need for the ecto trees to mine nitrogen from organic sources. Okay, so that's a lot. Let me just review a couple of these things here. So these AM dominant stands, they have this fast decaying litter. And so therefore, there's little need to invest below ground because there's a lot of nitrogen that's already in circulation. Relative to in the ectomycorrhizal stands, where their slow decaying litters are changing the dynamic such that there's a greater need to invest in their symbionts to help them get nitrogen from the soil, from organic matter. Now, this idea of different syndromes between the leaf litter and the root allocation, these are the data I presented to you are mostly from Indiana forests. We have gone on through some syntheses and meta-analysis and looked at this in other temperate forests. And there's a lot of other evidence, things I won't get to today. If you're interested in the references, I'll be happy to give you the actual references to this. But we see lots of differences between AM and ECM stands when they're occurring in the same landscape. So we're not comparing a tropical AM forest to a boreal ectoforest. We're looking at places in the temperate forest where both types of communities co-occur. And we see lots of differences in different attributes that we're interested in as ecosystem scientists. But really what we want to do, I started this talk by telling you that we want to look at ecosystem responses. We want to look at what does this mean for, this is a lot about nutrient cycling and maybe more than you wanted to think about the nitrogen cycle, but what does this mean about understanding ecosystem responses? So getting back to this question of how does this help us sort of understand the potential for these land ecosystems to be a carbon source or sink in the future. So before we get to that, there's one thing I just have to walk you through, which is that there's been a new paradigm shift in the organic matter literature of late, which this has happened maybe over the last decade or so, and it's totally different and it's kind of upended. It happened during the process of the time that we started our studies, and it's really helped us clarify our thinking about how some of these dynamics and feedbacks may play out. And that's that the idea is that the decay rates of litters of dead detritus has a direct effect on the type of organic matter that forms and its stability. Now, in the old view, the historical view was that if you had slow decaying litter, think of pine litter, think of your compost pile or something that you're familiar with, the leaves you're raking up in your yard, that if litter that just sticks around, decays slowly, oaks, lots of tannins, that's good for carbon storage because that stuff is still there. You still see the leaves. That must be good for carbon storage. Well, that's true to an extent. It's true for the amount of litter that's there. But in the soil, in the subsurface, most of the soil is not just what you see. Most of the soil is below that. In the soil, there's actually a new paradigm, which is that the stuff that decays fast, now think of the stuff that just disappears, the maple litter, that the microorganisms responsible for that decay turn over. And then the dead bodies of those microorganisms, or the necromass, those microbial products, actually stick to soil minerals and become very stable. And some of the most stable carbon that we have in the soil is stuff that it could be a sugar, a carbohydrate, something that would easily break down, but it won't break down because it's stuck to a mineral surface. And so just to conceptualize that with this little figure, think of a leaf. It gets decayed by a group of microorganisms. And then it sticks to this mineral, and this little thing that looks like a guitar pick is just representing mineral-associated organic matter, or MAOM. So that's the stuff that's in the soil that potentially could last for decades or longer. So that's why I have this little lockbox here. These are microbial residues that are protected from decay because of the way that they're adhering to mineral surfaces. Now that changed a lot because, again, it sort of flipped it on its head how we think about organic matter storage. I'm going to, in a minute, bring that into the main framework of how that's informed our thinking. So the idea is, I already told you that AM leaves decay a lot faster than ECM leaves. So one prediction would be that if you decayed both of them in a common environment, ECM leaves, you'd produce more stable organic matter. So we did that using an isotopic end member mixing analysis. We used soil that was developed under corn, which had a unique del13C signature because of its different photosynthetic pathway than C3 plants. So that just these soils just have different natural abundance values. And then basically you can measure the amount of new organic matter that forms if you have, we have plants that are from here, we decay in a corn soil, and we can actually use this end member mixing analysis to look at the new formation of organic matter. It's a really powerful way of looking at organic matter that normally would take us decades to study. So what we found, we decayed those AM and ECM litters, and maybe not surprisingly, those fast decaying AM litters, this is just a litter quality index that has lignin and nitrogen. It's basically, you can think of this as fast litter decay. All the AM trees that decayed fast produced more mineral-associated organic matter. This is just in 85 days, about 10% of the organic matter, that's this mayam, was produced in less than a year. So this stuff can form really fast, and that was pretty illuminating to us. Now, one sort of paradox with that, so that means that AM plants, I told you they accelerate nitrogen cycling more quickly, but they also are having, so think about that. They have AM leaves are decaying more quickly. So there's a lot of nitrogen in soil. So there's a lot of available N here. And potentially that's going to produce a lot of stable organic matter because they decay fast, and that mineral associated organic matter, that's going to be protected. But that's potentially, so that's represented here. But also when you have a lot of, I also said there's a lot of nitrate that's being produced. And so you have a lot of leaching losses and a lot of gaseous losses too. So there's just something fundamentally different that's happening in these AM systems where they're producing a lot of nitrogen. Some of it's being stabilized, some of it's being lost by leaching, and we're not sure sort of how that all shakes out in the environment, but it's fundamentally different than what's happening in the ectomycorrhizal systems where the litter decays slowly and does not form a lot of this mineral-associated organic matter and has very little nitrate production. So we did it, we wanted to try to play this out in the forest. So Adrienne Keller was a grad student in the lab. And so she did that same C3, C4 pairing. She took corn soil and she put them in root ingrowth cores. So root ingrowth cores are basically, you fill a core full of soil, you put it out in the ground, you see what grows in, and then you can start to look at the differences in root production. But because we put in that corn soil, we did this out in the forest at 3 different sites in AM and ECM forests, we could see how much of that mineral-associated organic matter develops over time. And what Adrienne found is that the AM plots, the ones that were dominated by AM trees, form much more of this stable organic matter. So red here, this is Harvard Forest, Leadicke Woods in Indiana, Cirque in Maryland. And so again, this is in the field, mature trees, they're forming more of this stable organic matter. So as opposed to the last study, which was a jar incubation, this was in the field. Now, we wanted to look at this, what this means. We wanted to do some more, investigate more sites. So this is, let me just walk you through this. This is a map. This is not a map of just some nice color coded. This is the AM tree dominance of the United States based on the US Forest Service has about 100,000 of these forest inventory plots. So we took all of that data, we classified the trees as AM and ECM, and then you can see ECM trees dominate sort of the Intermountain West and the Pacific Northwest, coastal California. AM trees are dominant here down in Texas, sort of mesquite and some others. And then mostly in the eastern, it's sort of this mix, this greenish color, where you have AM and ECM tree dominance. Well, we were mostly focusing on eastern forests, where we had those mixed types. Ashley Lang was a former postdoc, and Ashley went to these different sites, and she looked at a gradient of AM to ECM trees, and she found 7 sites. These were in the NEON network, and she basically found 6 to 7 plots at each site, which had either were AM dominated or ECM dominant. That's just sort of the relative abundance of the trees based on basal area. So you can see that these little steps just indicate they're about evenly dominated by AM and ECM trees. And it actually found that she measured that mayam pool, that stable organic matter pool, that at 6 of the 7 sites, they had positive slopes. So 6 of our 7 sites had this positive slope, meaning that as you increase the abundance of AM trees, we had more of this stable organic matter, more of this mineral associated organic nitrogen and carbon. One site, Lenore Landing, is a bottomland forest in Alabama. We're not entirely sure. This has very unique mineralogy, why that site was so different. But we're encouraged to see that when you go out and you look for these sort of syndromes, they are noticeable not just in our Indiana forests, but also across a wide range of forests across the eastern US. Okay, so that's a lot of information. What do we do with this information? So we're very interested in trying to make sense and put this stuff into context. If there's more stable organic matter, what kind of predictions can we make from this? If there's more stable organic matter that's being formed under AM trees than ECM trees, maybe knowledge of the type of trees that you have in a stand can tell us something about organic matter dynamics across a broad spatial scale. So we did this work using the Forest Service, about 2,000 of these plots we got soil samples from and looked at the carbon and nitrogen in those samples. And there's a whole lot of plots that underlie these, thousands of dots. So I made it easy for you. This is just the trend line of the slope of those thousands of plots. And I categorize these by southeastern, midwestern plots, and northeastern plots. Again, these are thousands of different forest plots. And this is the stock of carbon down to 20 centimeters. And what we found is you increase the abundance of AM trees, you have more carbon. And as you increase the abundance of AM trees, you have more nitrogen. So that's pretty powerful. So without any other knowledge, other than just knowing the dominance of the tree types, we have some pretty good predictability for understanding carbon and nitrogen stocks in the soil. Another application that maybe is more, sort of to try to sort of understand what this means for the carbon sink is thinking about forest productivity. So now I mentioned that thousands of these plots have had soils taken from them. All of the plots, over 100,000, have looked at tree growth and productivity. So we can use the full data set of the Forest Service. So we calculated AM and ECM dominance and looked at growth intervals over time. And then we modeled the relationships of growth based on the different ecoregions. Let me walk you through this because it's a little confusing. So this one does have all the plot. These are all the data points here. And then these are the trend lines I'll explain in a second. So we divided the country up into 33 ecoregions. This is just what the Forest Service already had done this, based on sort of dominant cover types, mixed mesophytic hardwoods, sort of different types of eco, just a way of sort of blocking for some of the spatial variability that you have across the eastern and western United States. And within each ecoregion, we looked at the function of, as you increase the AM tree dominance, so it's just, these are ECM dominated plots, these are AM dominated plots. What was the shape of the productivity? Was it not significant? There's no relationship. Is it linear positive, meaning growth goes up as you get more AM plots? Is it linear negative? Is it concave positive or concave negative? And as you can see here, I'm just sort of using a lead, over 2/3 of our plots showed this concave negative, that's these red ones, that negative relationship. So what does that mean? Well, that means that plots that had AM and ECM trees tend to grow the most, tend to be the most productive. And that was kind of cool and not something we initially suspected, but it does tell us something potentially about niche partitioning in ecosystems, where if these syndromes are true, and we have pretty good evidence they're happening, and these AM and ECM trees are in a mixed plot, maybe they're not even necessarily competing in the same way that they might when they're in a plot dominated by their own type, because they're sort of having independent ways of accessing nutrients from the soil. So for thinking, all the people interested in planting trees, and there's abundant interest in this now, this gives us a clue on something that we might think about when we're planting both AM and ECM trees. If nothing else, that may lead to greater ecosystem carbon uptake and storage, or at least more productivity. The last application I want to tell you about is thinking about the future world, which is under elevated CO2. So when I met Greg Benito, Greg's here, back about 20 years ago when I was at Duke doing a postdoc, and I started working at this plot right up here. It's the Duke Forest, and this is an elevated CO2 experiment that ran for 14 or 15 years. And we use a technology called FACE, where you expose the trees to elevated levels of carbon dioxide for a long period of time and look at the ecosystem responses. This is a pine plantation right here, Duke Forest. This is Rhinelander, which is aspen mostly, but with some pine and, with some birch and maple. And then Oak Ridge was a sweet gum plantation. And when I started my postdoc, it was sort of toward the end of these face experiments. And some of these forests were showing sustained growth responses to CO2. They were continuing to grow better when you give them CO2. And that was true at the Duke Forest. It was true at the Rhinelander site, but it was not true at the Oak Ridge site. And I wasn't sure, but I thought, well, one thing that's actually interesting about these, it's not a very good sample size, but the Duke Forest pine trees associated with ectomycorrhizae, aspen are dual associating, but because they have birch there, and because of the work that was done there, most of them was about, the vast majority were ecto-associated. And in the Oak Ridge, they were only associated with AM fungi. But, as any grad student knows, an N of 1 is not sufficient. No one's going to believe you. So we started thinking about this in a more synthetic way. So Cesar Terrera came to a workshop where we talked about this, and we hatched this idea of let's do a multi-factor meta-analysis. So we gathered all the different CO2 experiments we could find, and we said, what factors best predict whether you're going to respond to elevated CO2 or not? And there were really only 3. The amount of CO2 that you were given, that's the delta CO2 up on the third top. No one mouse is, there it is. The availability of nitrogen in the soil, and then the mycorrhizal type, whether you're AM and ECM associated. So that was pretty cool. The ECM plants in this analysis ended up being about 30% greater, consistent with those face forests I showed you before. And the AM plants had little to no growth response. But we weren't totally satisfied because, I told you at the beginning of this talk, there's sort of carbon that's being stored above ground, but there's carbon that's being stored below ground. Are those related? Because if they are related, then that's good. We can learn, a lot from that. If they're not related or they're offsetting, that could be troubling. So we did a second meta-analysis about 5 years later. This time we had 108 studies we could look at, and we looked at both the plant and soil responses to elevated CO2. What we found that the ECM trees were able to sustain their biomass growth under elevated CO2. And the mechanism they did that was they accessed nitrogen from the mineral-associated organic matter pools, leading to a decrease in soil carbon. And let me walk you just through this because it's really, really interesting. So on this x-axis here, we have the CO2 effect on the plant biomass carbon. So the blue is ECM. So you can see a lot of these numbers are 20, 40, 60%. They grew bigger when exposed to CO2. Now, the y-axis is how they responded in terms of the soil carbon. Notice how almost all these blue dots are below 0, meaning that they put carbon into their biomass, but it came at a cost of the soil carbon. They were mining nutrients out of the organic matter, and so they were losing the soil carbon storage, but they were gaining carbon in their trees. The AM plots did almost the opposite. They were not able to gain much in response to the CO2, but they were building soil carbon in their soils. That's the y-axis here. Now, this trade-off, this sort of negative relationship, is not at all how the models are structured. So in this paper, we went through, I just said this, in the paper, we went and talked about how the CMIP5 predicts the relationship between CO2 effects on plant biomass and CO2 effects on soil carbon. It predicts linear positive, but it's actually sort of like an exponential negative. So we need to fix that in the models, because if plants are mining organic matter to get nutrients, and that's changing the carbon balance of the systems, we want to make sure we represent that faithfully in these models. Okay, so I'm just going to end with a couple quick slides about what we're doing now in our lab, and then I'd be happy and excited to answer questions. So one thing that we're really excited about looking at in the future is understanding whether these main dynamics are driven by the trees or the fungi. Maybe that's a question you had as you're listening to this talk. It's a question I invariably will get at some point during my seminar. We don't know. It could be a combination of the two. So we're looking at dual associating tree taxa to see if we can look at these syndromes. So for example, here's some work in the aspen parkland where we have a mycorrhizal gradient of populus. Populus associates with both AM and ECM fungi. And just some of our initial work where we looked at the percentage of roots that, where we had ECM, this is actually from fungal, from the soil, where fungal dominance, the ectomycorrhizae were most dominant. We had smaller numbers of pathogens and saprotrophs, which is actually consistent with what we see in the sort of main effects, which I think is real and tells us that the fungi, at least the mycorrhizae, are definitely structuring or influencing the structure of the broader fungal community. Again, we're holding plant constant, at least tree dominance constant in this environment. And then, of course, we want to look at the fungal traits and a suite of other things, because not all ECM fungi are created equal. And so not surprisingly, there's going to be differences in enzymatic capabilities, et cetera. The second thing we're trying to understand is how long main syndromes develop and how sensitive they are to change. So we did some modeling of this because it's hard to run experiments for a long period of time. So I worked with a modeler, Josh Fisher, developed this model, a plant allocation model called FUN, or basically it's an optimality model. And then we coupled it with another model I worked with, another postdoc, Ben Solomon. He developed a model called CORPS. We actually put the two together and we had a model called the FUN CORPS. I know, not so good, but plants and then soil, we're sort of matching these two state-of-the-art models together. And we did this really interesting exercise where we said, what happens if we take these ECM and AM soils and we just cut down the trees and we grow up the opposite type of trees in that forest? So here's your total carbon. It's ECM dominated. This is just the surface of the soil. Here it's dominated by AM. If you cut down an ECM forest, if you take your AM soils and you just replant it, this is just an experiment sort of in the model, and you run it out for 30, 50 years, after about 40 years or so of only planting that with ECM trees, you sort of get the ECM phenotype, right? It starts to look like an ECM plot. So there's a legacy of the soils, but you override it after about 40 years. When we did the opposite, when we took ECM soils and we plant them with AM trees, you have a different effect. So in about 30 years, you have the different effects. Now, this is just a thought exercise. We have no idea if this is right. So what we're trying to do is we're trying to test this out using these FIA. This is the map I was saying about the red and blue. This is not, data this is from the Forest Service. These are AM and ECM dominated plots. This is how the forests have changed over time in the last 30 years. You can see AM trees are increasing in dominance. So now we're going back to these plots and looking at over 30 years, do we see changes in soils from when soils were sampled in the 1980s and 90s relative to more recently? So we're looking at how long does it take the soils to change once the vegetation changes? We're also doing this with common gardens where we grow the trees in a single uniform garden soil type. And then we're doing this with, instead of having common gardens, we're actually using root ingrowth cores where we let roots grow into soils, AM and ECM roots grow in, and we can start to look at the degree to which roots and their associated fungi are influencing the dynamics of the cores over short windows of time. So we don't have to wait 5, 10, 20, 30 years. This is collaborative work with Chris and Sarah. And then lastly, we're looking at this, trying to go beyond temperate forests and look at how this, whether this work can apply to tropical forests. So interestingly, most tropical forests, I told you, are dominated by AM trees. That's the pie chart here is this sort of peach color. But if you go into Southeast Asia, there are a lot of plots that have ectomycorrhizal trees. And some of these diptera forests are really interesting, where they have ECM-dominated trees, AM-dominated understories. It's really weird to call this an understory. There's just something really tall about these trees. And Ashish Nambiar is a student of mine who's looking at these dynamics to see if these syndromes play out in tropical forests as well. Okay, so just to summarize, these plant-soil microbe interactions that differ between AM trees, that owing to variation in the trees, owing to variation in the fungi and the microorganisms, bacteria, all the microorganisms, clearly are leading to some kind of a biogeochemical syndrome, fundamental differences in the way nutrients are cycled in these sort of integrated phenotypes. And what I would say is that these relationships, between traits and trait microbe and trait soil organic matter are potentially useful lens for thinking about all of ecosystem responses to global change. If we can start to understand how these pieces fit together and not just measure the tree responses, not just measure the microbial responses, but to put them together into an integrated framework, we're going to have a much better ability to predict how ecosystems respond to global change. So I want to thank a number of people that were involved with this. I've highlighted some of them in the pictures. These are some of my main collaborators, a number of technicians, and obviously thanks to the National Science Foundation, Department of Energy, and Smithsonian's Forest Geo Network for helping us set up and establish these plots. And with that, I would be happy to take your questions. Thanks for being patient.
questions?
Yes.
When we talk about the AM trees and the ECM trees, like AM trees always have correlating and molecular. So may I say when I compare two plant material, if one has for like lignin, then I can predict probably it has like more molecule. This is the first question. The second is like when we compare the ECM and AM, it's always like AM trees have some more bacteria, like naturally contribute, bacterial community, nitrogen over. As that all because these trees have more available nitrogen over.
Yeah, so on the first question, the nitrogen and the lignin aren't always correlated, and that's actually why we often measure the lignin to nitrogen ratio. There's some plants that have a lot of lignin and have very little nitrogen, some that have a lot of lignin have some nitrogen. And the lignin to nitrogen is probably the best predictor we have of the decay rate of a leaf litter, is those two, because of people often use carbon to nitrogen, but carbon could be any kind of carbon. And so by having this sort of more recalcitrant, unhydrolyzed residue that's basically what lignin is, that slows decay, having those two together are usually the best way to make a proxy. So if you have a high lignin to nitrogen ratio tissue, you are more likely to have that sort of AM looking soil response, more nitrogen available in the soil because the litter is likely to decay more quickly. So I would say that's probably true, but it's not just the lignin or the nitrogen. To your second question, I think you're asking about whether, maybe can you just rephrase the second one?
All the AM trees use more microorganism activities than negative cycles, especially nitrification, nitrogen population. It's all because the plant has energy. That's the only
yeah, so they tend to be slightly more N-rich environments, but and there's clearly some filtering that goes on. I mentioned this idea of, is there nitrogen fixation different? Because that would explain sort of what caused this sort of nitrogen enrichment on the landscape. So I didn't get into it and talk about it, but there is some environmental filtering that goes on. So AM trees can establish anywhere. They often establish in places where there's slightly more nitrogen enrichment, and then they reinforce those syndromes through their litter and through those, trees are long-lived, and so you have that very strong reinforcement that happens over time. Because when we go to common gardens and we look at like 13 years into the common garden, the soil's the same to begin with. We plant AM and ECM trees, it takes time for these syndromes to develop. They're not immediately readily apparent. So some of it has to do with the environmental filtering, but a lot of it is probably, most of it I think is doing with reinforcement of over 80 plus years reinforcing those environments. Now, there's not necessarily more microorganism activity. I think it's just differences in the types of microbes that are sort of selected for under this landscape. We have fast decaying litters. I didn't talk about this, but the mycorrhizae don't protect the roots from pathogens very well in the AM systems. And this is some work that Andrew and Chris and I have been working on, some of the pathogen feedbacks to the plants. So there's a lot of other dimensions of things that sort of go beyond just looking at the soil microbiome, like the root microbiome is actually pretty different. But that's an interesting question for sure. Yes.
Do the AM versus ECM soils differ a lot in their pH, right? And do you think that plays a role in this?
100%. 100%. Yeah. So on most of our sites where we have the most striking differences, these sort of syndromes playing out, there's anywhere between 1 and 1.5 pH unit differences. So if it's 5 in the AM plots, it's 4 in the ECM plots. Now, those differences are mostly at the surface in the top 10 centimeters or so. And that has to do with probably that O horizon and the organic acid production that's happening as a result of those ECM, those slow decaying litters. But there's a cascade, we know microbial communities are structured in large part by soil pH. That plays a big role in influencing communities. So it's part of the syndrome. It's not always the driver, but it's definitely something that we've seen, and where we see the strongest differences, it often is in places where the pH change is most strongly different. We've done some work with pH where we sampled down once with 50 to 15 centimeters in soil, and we measured every centimeter how the pH changed you know, in an AM and ECM stand. And they're really different at the beginning. And by the time you get to about 10 centimeters, they're only about a third of a pH unit difference. And by 15 centimeters, they're almost the same. So it's a very much a superficial thing that's happening. So deeper in the soil, it's not explaining what's happening at depth, but certainly in the upper surface soils, which is often what we sample, that's having a big effect on these syndromes. Yeah. Yeah, Greg.
I know you're talking about forest here, but do you think the main model can be extrapolated to other AMF communities like prairies or?
I think it's an open question. I would love it. And sort of I purposely put this last take home point here because I do think it's an open question whether if you have imagine you have any system where you only have AM plants or you only have ECM trees. Does this framework hang up? Do these principles hold? If you have assume you have variation in your community and you have fast decaying litters and slow decaying litters, or you have acquisitive plants and some conservative plants, even within like a grassland community, does that produce the same types of dynamics? Now, it's not going to be as spatially separated because in a grassland community, there's much more intermixing. You're not going to have monodominance or mycorrhizal dominance in the same way. But I think it's a really interesting question asking these things about the way that the litter affects the soil organic matter and structures the microbial communities and feedbacks to the plant that one could ask if you have enough variation within your community. So imagine you have slow growing and fast growing AM plants growing together in some middle succession restoration context. Maybe you'd have some of these dynamics playing out and you'd be able to make some predictions about the kind of organic matter that's forming and whether that's constraining or feeding back to affect plant growth. Yeah, great question.
Okay, it's right on time, and let's give another hand.
Thank you.